CSPGs, and then bring up the information that the ECM affects oligodendrocytes and also microglia activity, and then tell you about how we are thinking about strategies to reduce the production of CSPGs that occur following neural injury. And then at the end, I'm going to introduce you to fibroblasts that come into CNS lesions that many of us, I think, don't really appreciate, but they enter CNS lesions and they are also major producers of ECM following injury. So the ECM is a large network of proteins or molecules that support, surround, they give structure to organs, to the central nervous system. They are also very important for signaling because they can anchor many molecules and provide signals to different cell types. Members of the extracellular matrix would include collagens, of which there are many different subtypes, laminins, also many different subtypes, proteoglycans, etc. And it has been estimated that there are about 300 bona fide core ECM molecules. So I think this is a nice way to think about the extracellular matrix. This is the idea of a former postdoctoral fellow, Samira, who is now an assistant professor at the University of Toronto. So she tells us that if, in considering the extracellular matrix, or to explain the extracellular matrix of the brain, it is easiest to think of the extracellular matrix as the soil of the brain, the soil that sustains the garden, and therefore the soil is important for all functions within the central nervous system, and there's disruption to the soil, there will be consequences to things that are growing in that garden. So that's the thinking about the extracellular matrix. Just going to try to move this to the side. Here we are. So in the central nervous system, the extracellular matrix is organized into three major compartments. This is in the uninjured adult central nervous system. First, there is this collection of ECM molecules magnified here that are outside blood vessels. They constitute the basement membrane. They're very important for integrity of the blood-brain barrier, diffusion of molecules, etc. And then there's also a collection of some ECM molecules around some populations of neurons in the central nervous system, referred to as perineuronal nets. These are important for the survival of that neuron and also for formation of learning and memory. And then around or in between cells are this collection of ECM molecules, and they're referred to as the interstitial extracellular matrix molecules. And members of, again, are shown down here. So you have, different types of extracellular matrix. You have type 4 collagens that is in basement membrane, but within the CNS parenchyma, a lot of hyaluronan, chondroitin sulfate, proteoglycans, tenascin C, etc. So, like I said, I'm going to pay more attention to the chondroitin sulfate proteoglycans. And again, trying to move the slide. Apologies. Here we are. All right, the chondroitin sulfate proteoglycans. The chondroitin sulfate proteoglycans, or CSPGs, have been known in the central nervous system for many years now. Although they are normal constituents, they accumulate at regions of injury. And very popularly known is this collection of CSPG in red around a lesion of traumatic spinal cord injury. So this has been researched on for many, many years and has been driving the field of CSPG and ECM research in the central nervous system. And what one appreciates is that in this cross-section of the spinal cord that is transected, the CSPGs accumulate and exons that are trying to regenerate cannot. So this is a barrier forming a scar-like matrix that prevents CNS regeneration. The non-permissive nature of extracellular matrix, CSPGs in particular, is shown on the right, where at the bottom you have neurons growing happily away, but when you plate them on a CSPG substrate, you see that these very few neurons adhere, and those that do not do not fare very well. And again, apologies with my AV here. All right. Trying now to project it. And I'm not getting to that side. Okay, all right. Here we are. Again, apologies. So again, coming back to the CSPGs, it is a broad family. A subset of the CSPGs is the lactocan binding CSPGs. And these are the members of the lactocan CSPGs. You have agrican, versicans, of which there are 4 different isoforms in the central nervous system, and then you have neurocan, brevican, and brevican. So members are defined by this protein core, of which are these repeating disaccharides of glucuronic acid and N-acetyl glucosamine. The repeating disaccharides is also known as glycosaminoglycans. Functional activity resides in these GAGs that are heavily sulfated as well, and depending on where the sulfation occurs, that can have different functions, but activity also resides in the protein core, which differentiates one lactocan CSPG member from the other. The degree of GAGs also makes them different. And again, I'm just having problems here with my advancement. All right. We became interested in lactocan CSPGs because we have been in MS lab for many years. And Ray Sobel, a neuropathologist at Stanford, described the deposition of these CSPG members in MS lesions. Here they're showing us the edge of a lesion that is expanding this way. And in this edge, they show us this brown deposition of these 3 lactocan CSPG members. At that time, it was known that the CSPGs were inhibitors of axonal regeneration. In MS, we are still not thinking about axonal regeneration, but we're thinking about remyelination. And so that led us to ask whether or not the CSPGs would inhibit oligodendrocytes and remyelination. And of course, many have already gone over this morning, and I see it on the abstract outside as well, that many of you work on remyelination, and you know the importance of remyelination, the restoration of conduction of impulses, recovery of functions at best, but more realistically is the concept that remyelination can protect the underlying compromised axons from degeneration because of the exchange of molecules. So therefore, this is a realistic goal in the field of multiple sclerosis to enable remyelination in order to prevent axonal degeneration and progression of disability, and hopefully someday down the road, the recovery of functions. So we are interested, therefore, in remyelination, and we ask whether or not the oligodendrocytes would be impaired by CSPGs. And working first with cells in culture, you see that when OPCs are grown, they do very well, but when plated on a CSPG substrate, very few cells attach. Of those that do, they don't develop morphologically. I think perhaps the best example of the inhibitory nature of these CSPGs can be shown in this work with stripe assays. So in red are stripes that are coated with your protein of interest, bovine serum albumin as a control, or CSPGs to our right. human OPCs are plated on, and you can see that on controlled BSA stripes, the cells do not care whether they are on non-BSA, which is polyl lysine, or BSA containing area. But on a CSPG test, you see that these OPCs avoid the CSPGs very avidly. So same number of cells in culture, same medium, but given the choice of substrate, they avoid CSPGs. So we then went on to test the role of CSPGs in vivo. And again, many of you know this, two different models that I'm going to show you work in experimental autoimmune, encephalomyelitis, and inflammatory model of MS, where animals become progressively disabled, and lysolecithin demyelination that you heard of this morning as well. We like this because this is very quick. There is pretty much immediate demyelination and loss of cells in that area of lysolecithin deposition within minutes. And then within the first few days, that area is quite bare, and now cells have to repopulate, and you have mobilization of oligodendrocyte precursor cells by day 5, if not earlier, remyelination beginning by day 10. So it's a quick model to look at whether or not your drug or agent of interest affects that process. So looking first at the accumulation of CSPGs, this work shows that CSPGs are deposited in lesions of EAE and MS. And of the lacticans CSPGs that I referred to you earlier on, the one that we keep implicating is Versican V1. So you also have, some change to Agrican, some change to Brevican, but mostly it's Versican V1 that accumulates in lesions of MS and its models. And you show this example here. You see this example here. This is spinal cord longitudinal section, an animal at peak EAE severity, looking now for where this area has a lot of immune cells, CD45 positive, and in this area, prominent Versican B1 deposition. Shall I admit these people? Okay, all right. And then here's a blow up where you have CD45 positive immune cells co-localizing with Versican V1. By in situ hybridization, we have defined that it is myeloid cells, IBA1 positive cells, that elevate Versican V1 transcript. You also see that in a lesion of MS. Here's actually the rim of a chronic active lesion with a lot of immune cells, and in this area, Versican V1 deposition. As well, when you look across a number of MS specimens and you ask whether there is a correlation between areas that contain Versican and areas that do not, trying to again move this forward. Yeah, I'm just not seeing the advance button here. Here we are. All right. Here. All right. So here is a correlation looking at plaques with low Versican V1 and high Versican V1 expression, looking at BCAS positive cells that Wendy showed us this morning, where these are pre-myelinating committed oligodendrocytes to myelination. And you can see that in areas of high Versican accumulation, this tends to be areas that are low in BCAS positive cells. While I focus your attention on the CSPGs, I just want to acknowledge that many others have worked on other ECM molecules and have shown that these are also elevated in lesions of MS, and that they also impair oligodendrocyte biology to various extent. And here's a listing of those. Besides impeding OPCs that I've shown you, the CSPGs are also pro-inflammatory. And I want to give you examples of that. First, I want to tell you about infiltration of immune cells across post-capillary vanules of the blood-brain barrier into the CNS parenchyma. We can visualize those in EAE or in MS tissue sections, where here's the lumen of a blood vessel. In the central nervous system, the post-capillary vanules have two basement membranes, and you can see them, the first one here and the second one here. Cells tend to be trapped first between the two basement membranes to form what are called perivascular cuffs. And it's only when they begin to express matrix metalloproteinases that they can break the second barrier to get them into the CNS parenchyma. So you can see this infiltration example, another example here. And what we observed was that versican V1, besides in active lesions or at the edge of a chronic active lesion, versican V1 is also expressed in these perivascular cuffs very prominently. Here again, in example from EAE, the two basement membranes, the perivascular area here, and this is where versican V1 accumulates. And when you look for the immune cells, they are here, and here's a high magnification view. This is also in MS as well, and active perivascular cuff immune cells are about to enter into the central nervous system, and in the perivascular cuff is where you see versican V1 expression. So is there a function? Can versican V1 or other CSPGs, if elevated, promote the activity and migration of cells into the central nervous system? And indeed, by addressing that, we find that the CSPGs in tissue culture are very potent inducers of a number of matrix metalloproteinases, enzymes that are important for trafficking of cells into the central nervous system. And for some of these MMP members, the CSPGs are even more potent than LPS. And so when you do a transmigration assay in tissue culture, where you ask immune cells to migrate from one area to the next, and you add CSPG to the mix, you find that there is increased transmigration of immune cells. And so in this work, we postulate that the CSPGs that are elevated in the perivascular cuffs by immune cells, particularly myeloid cells, help to give the immune cells additional boost by elevating MMPs and also cytokines that haven't shown you to help cross the blood-brain barrier into the CNS parenchyma. And if that is the case, inhibition of CSPG content should be able to stop or reduce transmigration of leukocytes. And I'll show you one set of those data afterwards. Furthermore, CSPGs are important and have roles in the polarization as well as the toxicity of T cells. So in this example, we have taken naive T cells and polarized them in different directions to form T helper 1 or T helper 17 pro-inflammatory cells, or Th2 or regulatory T cells. And we have simply added CSPGs to that environment. So a mixed CSPG containing that you buy commercially, containing versican V1, but also other lactican CSPGs or purified versican V1, both of which can increase the the frequency of Th17 cells that form. So very pro-inflammatory, besides helping immune cells cross the blood-brain barrier. They also have a role in polarization of T cells along a very pro-inflammatory Th17 pathway. Moreover, these cells, having been exposed to CSPGs or versican V1 in this particular case, are also more toxic to oligodendrocyte precursor cells. So in this work of real-time imaging, in which we are looking in green at all cells, and in red, the cells that have that are undergoing compromise or death. And I would say that these green cells are oligodendrocyte precursor cells. The presence of Th17 cells that have some cytotoxic besides pro-inflammatory potential will lead to some death of these OPCs over the course of time. But if these Th17 cells have been exposed to CSPGs of versican V1, you see that the death is promoted. These cells are even more toxic. So all of these leads us to contend that besides the direct effect on OPCs, the CSPGs may inhibit remyelination by promoting the T cell killing of OPCs. Just some ongoing work, just to briefly tell you some of the things that we're doing with CSPGs in inflammation. This is the work of a PhD student in which she has generated a versican flox mouse, across that to a CCR2 Cree-ER in order to, following tamoxifen induction, delete versican from CCR2 myeloid cells, monocytes in particular. When you do that and you induce EAE in these animals, in red, these animals don't develop EAE. So this is actually a very strong phenotype for Mariam, who is following this work in terms of trying to understand the mechanisms. So do CSPGs affect microglia biology of interest, particularly to me, but also to many of you? Here we are looking at a chronic active lesion of multiple sclerosis. It's chronic active because the immune cells are now at the rim and have dissipated from the center. And at the rim of this chronic active lesion, you see many CD45 positive immune cells in green, and these are in the vicinity of CSPG expressing cells, which we think are microglia macrophages. So there's close association. Well, first of all, there's elevation of still prominence of CSPGs, particularly versican V1 at the rim of these chronic active lesions. Should have mentioned that these chronic active lesions are very, very consequential in multiple sclerosis because their number correlates with progression of disability. So here we have CD45 positive immune cells, many of which are microglia macrophages, having shown you that data, correlating with the presence of CSPGs. And so we ask, what do CSPGs do to microglia? So unlike this example that I showed you earlier on, where the OPCs would avoid the CSPG stripe, when you put microglia on, microglia love the CSPG environment and they affix to that area. So we think that to some extent, this affinity may account for these correspondence of CD45 positive cells with CSPGs at the rim of chronic active lesions. And moreover, we think they are consequential, that they are neurotoxic. Here's is an example of such toxicity. So here the experiment is exposing macrophages or microglia to CSPGs or other ECM or controls, and then you take the supernatant and put on neurons. And you see that the supernatant from microglia or macrophages that have been exposed to CSPGs are very potent mediators of neuronal killing. So the CSPGs, we think, help account for neurotoxicity of CSPGs and perhaps for the particular toxic nature of the rim of chronic active lesions. So are CSPGs promoters of microglia toxicity and lesion expansion of MS? This is something that we are excited about. I put up this slide because this is a microglia session, just to remind us that there are many flavors, functional states of microglia. So rather than a yin and yang, good versus bad, this is just to depict that there are many functional states of microglia. And many of you are aware that these functional states are dictated by the proteins that they make, their transcriptomes, etc. These are some of the molecules that sway them towards degeneration associated. And we would like to put forth the idea that the extracellular matrix, such as members of the CSPG families, are critical mediator of this switch, particularly to degeneration-associated phenotype work in progress. So at this point, just a reminder that challenges imposed by CSPGs would include the following: inhibition of axonal regeneration that's very well documented in the traumatic spinal cord injury field. Haven't shown you this data by others also in the traumatic spinal cord injury field that CSPGs are important for, will attenuate the proliferation and migration of neural stem cells and their progenies. I showed you some data that they might prevent remyelination, promote neuroinflammation, increases microglia toxicity. So therefore, I think we need to interfere with these CSPGs. So normally the CSPGs are present within the central nervous system, but they are substantially elevated in lesions in the central nervous system. So we think where they become undesirable and we think that they need to be interfered with. Others are thinking along the same lines. They are using antagonists to receptors that CSPGs interact with. One such receptor is PTP sigma, and their efforts to use peptides that inhibit PTP sigma signaling in the traumatic spinal cord injury field, some of which have reached clinical trials. And then there's also the work on using chondroitinase ABC, an enzyme to remove the glycosaminoglycans of CSPGs. This, in the traumatic spinal cord injury field, is locally deposited to remove the GAGs, but the core protein still remains, and that also still has undesirable roles. And also, when thinking about multiple sclerosis with multifocal lesions, it is not practical to micro-inject chondroitinase ABC to multiple sites. And so therefore, we took a different approach to prevent the production of CSPGs after injury, particularly to take advantage of the injury-enhanced elevation of CSPG synthesis after injury. So how do we go about preventing the production of CSPGs after injury? And here, we went back to biochemistry 101 to try to understand how CSPGs are made. So here is the protein core of this serine residue and a linker region. You have this repeating disaccharide of glucuronic acid and N-acetyl glucosamine, which itself is formed from N-acetyl galactosamine. Sorry, I got that reversed. It's N-acetyl. In CSPGs, the GAGs is N-acetyl galactosamine, which itself is formed from N-acetyl glucosamine by this 4-epimerase enzyme. So the approach that we took was simply to use a false substrate. So we have generated this molecule. This is fluorinated at the 4 position. We tested many small molecules before arriving at this. So this is fluorinated at the 4 position. And if it is acetylated, per-acetylated, to facilitate entry into cells, this becomes per-acetylated 4-fluoro N-acetyl glucosamine, which is a mouthful, and so we have simply called this fluorosamine. So to test fluorosamine in vivo, we started with the EAE model, where, if you recall, CSPGs are at the perivascular cuffs that we think help with the migration of cells across the blood-brain barrier. So in this experiment, we are using EAE, and we are initiating treatment at onset of clinical signs, where immune cells are beginning to enter into the CNS parenchyma from perivascular cuffs. So in that context, when treatment is started here with daily treatment thereafter, just over a few days, you have diminution of clinical disability. And if you look into the spinal cord for histology, you see that these perivascular cuffs, immune cells have migrated into the CNS parenchyma. And in the context of furosemide treatment, we have reduced that transmigration, consistent with CSPGs facilitating immune cell trafficking into the central nervous system. In the lysolecithin demyelination model, we have also used furosemide. Treatment is initiated 48 hours following demyelination when CSPGs, particularly Versican V1, is beginning to accumulate. And daily treatment, here animals were killed at 7 days following demyelination, looking into the lesion. And you have seen examples of this already presented, where OLIG2 is a transcription factor that defines cells of the oligodendrocyte lineage. If they are PDGF R-alpha positive, this is an OPC, CC1 positive, a mature oligodendrocyte. And you see that treatment from day 2 to 7 following lysolecithin demyelination, in this case, promotes the number of mature oligodendrocytes that form. We think consistent with the idea that an inhibitor, the production of an inhibitor has been reduced. And looking at myelin reformation using EM and this quite crude score, we got a value of P0.05, just significant in the case of furosemide treatment for profiles of remyelination. So this P0.05 isn't good enough. So we went back to our chemistry collaborator, CC Ling, and asked for more compounds to try to improve upon efficacy. So here is furosemide at the top. So again, many different derivatives were made, one of which is this compound with 2 fluoro at the 4th position rather than 1. And this compound in red, very, very effective in reducing the synthesis of CSPGs. In this case, in our bioassay of cells making CSPGs in culture. So we have now difurosemide, 2 fluoro, and this is what happens when you treat EAE animals with difurosemide. Treatment initiated here when animals are quite disabled, and very quickly you see this resolution of disability. And when animals are killed at this time point at the end, looking into the spinal cord, the difurosemide treated animals have not much Versican V1 or CSPG, consistent with having reduced the production and subsequent accumulation in lesion compared to the EAE control. And now looking again at regenerative profiles, oligodendrogenesis being increased by difurosemide, and looking at the production of new oligodendrocytes and myelin profiles using this transgenic line, you see that in difurosemide treated animals, the cells tend to have very, very prominent processes compared to EAE control. And even in this area of inflammation, with many of these dots being immune cells, in the case of the difurosemide treated animals, the oligodendrocytes are regenerating. So we think this is of interest. We continue this work, now moving on to what we call our 3rd generation compound. We have created this difuroseminol with this OH group here. And there are many advantages, including better conjugation properties to a tracker dye, putting on other analogs, other moieties, etc. And now we're beginning to use this intranasally. I see a number of groups here use the intranasal approach. We heard a presentation earlier on, and I see that in at least one of the posters out there as well, This is quite new to us, but I really like the intranasal idea. You have intranasal administration to facilitate access across the blood-brain barrier through the trigeminal and olfactory nerves. You can have spinal cord outcome as well, even though you have intranasal administration. And for us, we potentially reduce exposure in the periphery. And that's because even though CSPGs are turned over very slowly in the normal central nervous system, and where you have a window of opportunity after injury when a lot of CSPGs are made, to target the injury-induced CSPG synthesis. The CSPG synthesis occurring elsewhere as well, in joints, in the intestine, etc., that we need to avoid. And so this is our intranasal administration with a clinical effect in EAE, and also with evidence of reduction of CSPG accumulation in EAE-afflicted animals. So work continues along that line. So what are other ECM constituents in lesions of MS? And what do they do? So we are interested in this. We are focused on the CSPGs, others are focused on others, but we are quite confident that there are many more ECM members to be discovered, some that might even be more important than those that I've described. We are focusing on the rim of the chronic active lesion because the number of chronic active lesions as well as the activity in the rim of these chronic active lesions correlate with progression of MS disability. So here we are at the rim where we know that there is microglia, a lot of axonal degeneration, failure of remyelination, and comparing that to the core, and also normal appearing white matter and normal control. So this is our attempt now to do unbiased proteomics to try to identify the ECM. So we have, as an example, dissected these areas. We have run controls just to make sure that our areas are of particular types. So for instance, in normal appearing white matter, you still have PLP, but not so much at the rim or core. And the rim is where HLA-DR positive microglia macrophage would be. And then we bring this through fractionation with success, increasing strength and detergent in order to arrive at a pellet that is highly insoluble and enriched in ECM proteins. This is what we then bring all of this through shotgun proteomics. And this is an example of a run in which we have 87 bona fide ECM molecules that we have identified. And also we have examples like this in which here in the highly insoluble ECM enriched portion, you have this particular ECM that is more focused on the rim and a little bit in the core, but not in the normal appearing white matter. So these are the candidates that we are chasing after to try to look for new ECM that have not yet been researched on in the central nervous system and that may have very important functions. So this is in progress. Finally, in this last part, I want to tell you about fibroblasts. Fibroblasts are universal in us, but there are very few fibroblasts within the parenchyma of the central nervous system. But fibroblasts are present in the perivascular space. They're present at barriers such as the meninges. And if there's a breach of the meninges or impairment of the perivascular space, fibroblasts can potentially infiltrate into the central nervous system parenchyma. So here is a lysolecithin injury, 7 days following injury, looking at fibroblasts by virtue of expressing PDGF-R beta, but also collagen 1A1, the activated form of fibroblast alpha smooth muscle actin, and also these two other ECM molecules, and a number of other markers, and the absence of others that now lead us to think that these are fibroblasts. So they are in lysolecithin lesion. Here's the blow up. They are also in EAE lesion, and you can also see these in lesions of MS as well. So they infiltrate into the CNS parenchyma. In the lysolecithin lesion injury, and this is at 2 weeks following, in which there's now a lot of reparative oligodendrocytes. So within the lesion itself, you see oligodendrocytes, but also in red fibroblasts. So they are there in the lesion by spatial transcriptomics. And here are some of the markers, PDGF-R beta, a number of collagens, and then down here, vitronectin, dicorin, etc. They are factories of extracellular matrix molecules. And because we have spatial transcriptomics, we can do things like cell-cell interaction, in this case using cell chat analysis, and show that these cells, fibroblasts, talk to oligodendrocyte precursor cells, talk to microglia, macrophages, and what are some of the signals, etc. So they have the means to affect the lesion as the lesion evolves or attempts to repair. We think that these fibroblasts are inhibitory for remyelination for the following reason. Where they accumulate, so here is a lysolecithin lesion, PDGF-R beta positive cells here. The OLEC2 positive cells are outside of that red scar that the fibroblasts form, and the quantitation is shown down here. And in tissue culture, you can show this quite nicely. OPCs growing alone and a higher magnification. And now when you put down a fibroblast monolayer in white, these OPCs avoid the fibroblast monolayer and they hang out in areas where there are no fibroblasts, and they don't do as well as their controls without fibroblasts. They are also present in multiple sclerosis lesions. There's a very famous database generated by Absinta and Danny Wright, published in Nature, of different types of MS lesions. And we have gone in, reanalyzed to try to get at the fibroblast population. So they're there, not that many in this particular case. Sometimes it's because of extraction method. But you see that the transcripts that they express, such as PDGF-R beta, a number of ECM molecules, they are there in MS lesions. So far, we have defined two different populations, 0 and 1, where one of this is has signs of cells undergoing apoptosis, but the majority of the fibroblasts are at the edge of a chronic lesion, in the lesion core, and not in the normal appearing of periplaque just outside of a plaque area. So they are there in MS lesions. We think that they are important factories of deposition of yet other ECM molecules, particularly the collagens. And our experimental data in animals tells us that where they accumulate, these are areas with fewer OPCs, and therefore fewer but lower potential of regeneration. So I come to the end and some summary slides just to remind you that the extracellular matrix, members of the extracellular matrix, can directly impair oligodendrocyte biology, but there are also indirect effects through the ECM engaging immune cells that then affect oligodendrocytes, such as the Th17 killing of oligodendrocyte precursor cells, and that many more alterations of the ECM remain to be discovered in neurological conditions. And with regards to the challenges that are imposed by CSPGs, we would propose that the furosemins, by having a window of opportunity to inhibit the injury enhanced production of CSPGs, may be able to reduce the subsequent accumulation of CSPGs in CNS lesions following injury. So the flow then, hopefully you are introduced, you're reminded of the extracellular matrix, a subfamily, the chondroitin sulfate protoglycans, that they affect oligodendrocytes and also microglia. That we have used furosemins to try to lower CSPGs, and that fibroblasts are something to contend with in lesions of the central nervous system, and they bring with them many ECM members that are deposited. So with that, just want to acknowledge funding sources, the people in the lab, and collaborators. Thank you very much.
Thank you very much, Gui. Wonderful talk, of course, as always. So we have question for Dr. Gui. Pablo Lopez.
And then when certain cell, new cell, that express normally one sigma receptor and the one that can express an inflammatory.
So the reason we use BSA in that experiment was because this was a very early experiment, and the reviewers let us get away with that. But these days we have used more equivalent controls like heparin sulfate protoglycans, or polyarginine plus, etc. PTP sigma. I don't know about PTP sigma expression outside of axons. There's likely that literature, but I haven't followed that literature. PTP sigma in that particular case is in the context of neurons expressing PTP sigma, where that interaction with CSPGs was identified as an important mechanism by which CSPGs were inhibiting axonal regrowth. So that's the context. PTP sigma on immune cells, likely, I just don't know that literature. It's a great question. First of all, the CSPGs are very important during development by forming boundaries early on in development. And then in the adult central nervous system, they're still there with many important functions. Following an injury, the CSPGs are likely trying to form barriers as well. So in our lysolecithin lesion experiments, we have always used, we think it's important when CSPG treatment is initiated. We have always started treatment after the injury has formed, and at a time period in which CSPG deposition is beginning, rather than right at the beginning. Some years ago, Michael Schwartz's group used a different approach. They used a xyloside, a xylose-containing compound, for CSPGs of the protein core, there's a linker region, the first residue of which is xylose. So they use a xyloside to try to interfere with that xylose. And in traumatic spinal cord injury, when they use xyloside, immediately following traumatic spinal cord injury, the outcome was worse. And if the xyloside treatment is started a few days later, then the resulting functional recovery from traumatic spinal cord injury is better. So I think there's a very important role for CSPGs as barrier molecules that we have to contend with. Therefore speak of the window of opportunity that that you need to know when the CSPGs are being made.
In your flows, you say that the axon is not regenerated. Is due to something special of the proteoglycan on the axon, or is due to the effect of the proteoglycan on the oligodendroglial cells? This is the first question. And the other question is, since in the model of you use CIA8 as a model, but in the cuprison model, the oligodendrocytes are destroyed. Have you tried in this model the effect of the fluoramina, how is, or you have not the idea?
Yeah, we haven't used the Cuprizone model at all. It's a very good point. And then I have to apologize. I didn't sleep enough last night. Tried to catch a nap after lunch, but was woken up by a security guard. What was the first question again? I'm sorry. Can you repeat the first question? Yeah.
Consequence of the effect of the proteoglycan on the oligodendroglial.
Yeah, the effect of proteoglycans on axons versus oligodendrocytes versus immune cells. I think all of these need to be, all of these are possible. What we find though, with in the lysolecithin model is that the elevation of versican B1 and also in EAE is only on myeloid cells, the injury-induced elevation. So if oligodendrocytes are making these proteoglycans, it doesn't seem to be very substantial. So in that context, we think we are affecting CSPG production that is elevated in the myeloid population, immune cells.
You showed that the microglia with the species what's going on there would be the mediator and what was the phenomenon? Are they dying by apoptosis or something like that?
Yeah, we don't know. Yeah, no, we don't know much at this point, but we're going down that direction though. So we're trying to make sure we have a serum-free defined medium environment that reproducibly produce that phenotype and then subject that to mass spectrometry. At this point, we don't know how the neurons are dying other than they die.
Thank you for the talk. So in vivo, you see that these proteins are located at some spot, forming barriers and so on. But then in culture, you put them soluble, right? So the question is there a soluble form in the animal, in the lesions that you could think, I don't know, maybe crazy, but to think of it as a chemokine or maybe as a marker for injuries?
So it's a great question that has been raised. So in vivo, we document all of this, and then we try to model this in vitro. So how do you present extracellular matrix molecules to cells? That's a very consequential question. Presently, we just adhere CSPGs down, recognizing that it may not actually be the right orientation that cells see in vivo, or we actually also add that to conditioned medium to the medium as a soluble factor. We are comfortable with the soluble form because for versican in our proteomic analysis of MS lesion, we see that versican in MS lesion is present in the insoluble ECM, but also increasingly in the soluble fractions as well. So some ECM molecules, presumably because of proteases that are also in lesions, become more soluble. And so you can present these in the soluble form. But at the end of the day, it's not an easy experiment to do because the cells may not be, in vivo, would be seeing a mixture of ECM molecules all interacting with one another. Here you are just adhering down. That's right. And then you need to find corroboration in vivo. Yeah.
That's very short question.
Okay, I was seeing the stripe assay, just a curiosity question, with the CSPG. The OPCs seems to interact more each other, like to form bulks. Is that just an in vitro effect, or do you see that OPCs interact more in the presence of the CSPG?
Yeah, I think it's probably an in vitro artifact. When cells can't go elsewhere because they are constrained by the barrier, they tend to aggregate. If you culture at very, very low density, you don't see that effect. Yeah.
Okay, thank you very much. We really appreciate your talk. We